So not this part's not so important to record. I want to capture all your sharing. So anyway, that's there as we're going forward. These resources, and then remember we've got the sort of shared folder there as well. I think this. So with that, then, any questions about sort of the timeline? Oh, one other thing is, traditionally we have a webinar in the first 2 week-ish of December. I haven't double checked with Susan about specific dates yet, but one of the things that we need to provide you some training on is, so how do I publish my lesson to Cubes? And how do I interact with sort of the Cubes portal to sort of get my lessons up there? And as your final revisions are coming online, that's something that you'll be wanting to think about. And so we'll be reaching out once I've sort of coordinated with Susan and myself and the rest of the team to deliver that webinar usually happens in the first part of December. To make sure everybody has the knowledge they need to sort of move forward toward the publication phase as we move into that. So with that said, any sort of quick questions about the timeline or process for sort of moving towards that publication and presentation of lessons?
Is
All right.
there,
Yep.
James, is there a new folder to upload the revised drafts?
Yeah, I'll put a folder
Okay.
up. I haven't shared it yet, but I will be sure to share that. All right. Well then, the main agenda, the main thing for today is for you guys to be reporting back from your what was it like to pilot your lessons? And this is pretty informal. It's really just a chance to kind of share out what it was like, but also maybe like, well, what did you learn from actually taking that lesson that was a paper, something written in paper, and turning it into actual something tested in the field. And so some of you have chosen to share slides, and so I'm just going to transition into the slides here. I think this is um, probably one of your slides, but it doesn't have anybody's name on it. So let's see. Exploring.
That's mine, James.
Okay, there
Yeah.
we go. Well, guess
Yeah.
what? You are up.
Yeah.
So,
So, oh, sorry,
tell
James,
us
go
what you
ahead.
did. Yeah, go ahead.
So my lesson was exploring riverine food webs, and I was able to pilot it in two different universities, where the goal was to have students collect and identify basal resources, macroinvertebrates, and fish using morphology and functional feeding groups, and then create a pre and post river food web with that. So this was piloted in mid and late October. But the where is the Rabbit River in Michigan, which is in the west side of Michigan, as well as Sugar Creek in Indiana. And the students were Hope College students in an aquatic ecosystems class, and I think there were 27 students in that class. And then Wabash College advanced ecology class with Brad. I had 13 in the one I had done for the field lab, and then he also did it again with another lab he had. So these were more upper level students who had some knowledge of food webs. Both courses, I believe, didn't have like extensive knowledge of the food webs previously or work specifically on rivers. The biggest challenges I found was not having worked with these students before. I wasn't sure of their background specifically, so understanding like how much they already know, like the modes of work that they do in the course and the group dynamics was a big challenge to like just come in and figure it out on the fly. Um, as well as providing the background students needed to complete the field lesson. So when I was at Hope College, I was able to give an introductory lesson the day before we went in the field about field safety, trophic levels, the functional morphology of invertebrates and fish, thinking about like mouth placement, different body parts, and how that reflects the fish and the invertebrate feeding groups. And those students were identifying organisms based on those morphological traits in the field, which I think worked really well. But we had less of that in background information for the second time around. So I think I'm incorporating that into the lesson to make sure that that information is front and centered and given
Mhm.
prior to going out in the field for students. And then the next slide, James. So I asked students before anything to go over and create a river food web of what they thought. So that's on the right here is the before picture of one group. So they had
Oh,
like
that's cool.
fish, plankton, and whatnot. After this field lesson, where the learning objectives were to use routine sampling equipment to collect invertebrates and fish, identify these aquatic invertebrates and fish based on their trophic groups and morphology, and then construct these realistic riverine food webs using at 5 different taxa, which they clearly did a lot more. But the after picture as well as the standalone picture were after the field. This is what the students were drawing. They were much more specific, much more detailed. They were including the organisms we found in the field those days, and they were much more complex and realistic.
Hmm, definitely. I love those pictures. And so I think you maybe have the record for testing it against the most different classes. I don't know, and taking it on the road as well, which is pretty impressive.
It was a
Uh,
lot of fun.
yeah, yeah, yeah. And it's interesting because you also probably got to have some of the same challenges that kind of the first few cohorts of scholars had in a sense of testing it against a group of students that you didn't necessarily have a lot of previous background with. And so you essentially do this with two different groups, one of which you got to provide some background and one of which was, sounds like less so. Did that, but it sounds like that was able to, you helped you inform what you would, some of the materials you might want to add into the lesson given those two different scenarios.
Yeah, for sure. It helped me like identify what was needed and like what's more optional for those field lessons as well.
Very cool. So what would you say would be the main take home of like like sort of just the lesson in your head versus the lesson now that you've tried it in two different audiences?
I think my main objective was for other than like the learning objectives here were students to get outside, get comfortable, and just see what's in the field. And from the reviews and from some of the assignments I've seen for the post assignments, students were like, I've never known there was so much diversity in these small streams. I never knew anything about aquatic insects or like how many there are. And it just made me feel really good about this lesson that like students are getting out of it, that these things are complex and they didn't know that much information beforehand, but now they know a lot more about the place they went into. And that made me feel really, really good about that. And that was an outcome I was not quite expecting coming from this, is just like the breadth of knowledge and care that they now have about the creeks and rivers that we were working with.
Well, that's pretty cool. Any other questions from the other folks? All right. Thanks for sharing. Well, this would be like if this was an assignment in my class, it'd be like you didn't put your names on it. So minus 5 points. Let's see.
That's
Photographic
me.
scavenger hunt.
Yes, that's me, Mari.
Okay, sorry, I can't see you, Mari. Go for it, Mari.
Okay, yeah, sorry, I'm in like in between meetings, so I'm in a coffee shop corner on the floor. So if there's background noise, I apologize.
No worries. I'm in a similar
Uh,
situation.
yeah. So I feel like I'm the exact opposite of Shaylee, where I piloted this with 27 a class environmental studies 300, which is 27 students in a service learning course. They're all majors or environmental studies minors, a couple biology students, but I know these students well. We spend 3 hours a day already in the semester when I ran it, and so I feel like my challenge is going to be the exact opposite of like trying to scaffold this down for my freshman level or non-majors, or thinking about the cubes lessons for a class that has zero experience. So
Mm,
what we did for this class was the idea was to use photography and to go out in the river and try to use that as a tool for observation and storytelling on our urban river that fronts our campus, and give the students some background information, terminology, concepts, basic concepts in river ecology and hydrology and some of those terms, because they're environmental studies students, so they may not have had much exposure. Some will, some won't, depending on their minors. Yeah, we piloted this. The students had a bunch of content already going into it. We'd already talked about tragedy of the commons, values of rivers, the history of this river. We looked at management plan for this river. And they'd already kind of learned about rivers and some key terms. So they came in with a lot of content knowledge, which was super helpful. The first thing we did was to look at some historic photos of the Boise River over time. And they got to see how it's changed due to different urban activities. And so they got to interact with those photos and kind of look at what had been. This is a working river. We've got dam, we've had major channelization, we have a flood, we have canals, we have agriculture, we have, gosh, all kinds of different examples of how this river's been changed over time. So it's a good river to look at. I even showed them that picture on the screen there. That was the campus, where you can see it used to have 2 channels of the river, and now they filled it in. Now that's actually the campus. So that was a fun picture to find.
That is cool.
Yeah, it's kind of fun to look at the historic photos. After we did that, the students had been working with the management plan for the Boise River already, so they were pretty familiar with it. So I divided them into 4 different teams. And they went and looked, they took pictures and went and looked at alongside the river. They went and looked for examples. So what I did was have them go into I had them go in 4 different teams. So one team was just looking at sort of channelization, erosion, geomorphology changes. One was looking at the riparian habitat. One was looking at water quality, and one was looking at kind of aquatics and fisheries. And they took pictures of those. They drew some maps too of the river ecosystem things they noticed. There's a picture at the bottom. There's a person fishing, which is kind of cool, during class. And then this one of the pictures is a beaver wrapping. This class actually did that last year because those are large stand cottonwoods that we have along the river that are kind of keeping it in place. There's some construction right on campus. They took some pictures of erosion. They got some pictures of wood because they move it out for river floating every year. So there's no wood in the system. There's pictures. I think one of the pictures is actually, some homeless encampment, right? So there's one on the bottom, and then there's one where they're actually standing in the spring flood zone. So it's the river changes a lot in the spring when we have our dam release flows. So they're actually standing under the bridge there. So it was really fun. They came back, each team shared their pictures, and they all had kind of a shared doc. So they went ahead and wrote down what the other group observed in that category, and it worked great. The only thing I would say was really challenging was I tried to do too much. So I was like, oh, this is prep for their service learning field days. I'm also going to have them use iNaturalist and Merlin and do some of that and some field journaling. And so I just tried to put too much in, and they came back and they're like, that part was just not good. We didn't know how to use the apps, and we didn't really know. So I think if I did it again, I would absolutely cut that part out and separate it into a different training day for that. And then I think the biggest, like I mentioned, the biggest thing for me is just thinking about how I would kind of change this to make this more generalized for a freshman intro global class.
Very cool. And and then Nancy, because you're going to pilot this in a different river system, right?
Yeah, on the Salmon River in Oregon, so it'll not be quite as urban, even though are just as urban rivers. I mean, it's urban enough, but it's not. We talked about doing it in Willamette because I'm at PSU, but I have a teacher, a K-12 teacher, high school teacher, who's going to be taking it to the Salmon River with his students on the 18th. So I'll have more to report, but 2 very different types of students. I don't know if he'll get the students I'm sure he'll get feedback from the students, but I don't know if how much feedback a high school group will give. Hopefully that'll be helpful. But I also have a couple teachers just kind of going through it. I have another teacher who's going to test it, but it's not till much later. So that will just be an extra bonus down the road, and hopefully it won't result in too many changes because she's not able to do it. That's the challenge I have with kind of that K-12 ecosystem is when they can do it based on what they are teaching. But we do have one coming up soon, so we'll have a little bit more before we're turning in the final lesson.
Great. Yeah, and it strikes me just kind of seeing Mari's photographs here that piloting it, one of the things that, obviously, keep, use protecting people's privacy and so forth. But some of these images that you guys have generated from the piloting could potentially serve to illustrate the lesson itself. Like here's an example of something that, students found in these different categories, and just to kind of help bring the lesson to life with some actually real world examples. And since you're doing it from multiple rivers, I think that could be actually quite cool if you sort of have those elements of the different river systems reflected. Like here's an example of, a human impact from one river versus another as you're kind of handing it off to somebody else. It's already been tested on multiple river systems, so that will help make it generalizable to someone who's coming at it from, Illinois or Oklahoma or some other place where the rivers would be different.
Right. Yeah,
so
good
that's pretty
idea
cool.
on the photos, incorporating more photos.
Great.
Other thoughts, questions for these two? All right, awesome. Thanks for sharing. Who we got next here? Let's see. Oh, I think this might be John.
Yeah, that it
Again,
is.
no
Yeah.
name.
Yeah, sorry. You didn't list name as a thing to put on it, so I thought it'd be obvious in this case. Yeah, so yeah, we did a pilot of my project looking at surveying amphibian communities along rivers. So I took my Bio 252 lab course, which is sophomores. They're bio majors. The majority are pre-med or interested in going in that direction, but we have a whole array of different folks with different interests across biology. Yeah, I took all three sections out to a backwater marsh called Banner Marsh that is about 25 minutes from campus. That is actually a reclaimed mining site where students could do the amphibian survey work. And when we got out there, I had them get into groups and come up with hypotheses for how different environmental conditions would affect the amphibian communities there. And then at least my goal was for them to collect some data to start to address that hypothesis. We only had one lab period for at least for this initial pilot. So I was like pretty clear that we may not be able to fully address it, but at least start getting there. So they collected some data both on the amphibians. We did a visual survey where they would work with their group to try to count and identify the different amphibians they saw. And here it's pretty much frogs. There's not a lot of salamanders in central Illinois. And then they were supposed to visually represent their data in some kind of graphical format, and then see how that relates to their hypothesis and discuss what next steps they would do. Yeah, I think it went pretty well. I did, think I tried to do too much. So this was our third class period. So while we were out there, I also wanted to show them some of the different ways to quantify amphibians. So some of them got a little bit of experience doing DipNet surveys where you'd use to quantify like larval amphibians, and we did that a little bit. But others, were doing water quality sampling too. I wanted to get in there too, and it's like, okay, this is probably too much for one single lab period. So I mean, they got a taste of that stuff, which is really all I really wanted, I think, because this is kind of their first field lab experience. I mean, we did one other one in a local park, but this was our first one where we actually went to a site a bit further away and got out there. So I think part of it was just getting them that field experience and exposing them to aquatic systems in general. But yeah, I mean, I thought they enjoyed it, and I think the product, they did a worksheet where they had the graphs and they had the reflections and insights, and I thought those look pretty good for the first trial of this lesson. So yeah, I was pretty happy with it. And I think just thinking about, and this is something that came up in the feedback I got too, like structuring how much time to devote to each thing and making sure that I have that planned out in the lesson for going into cubes would be good moving forward.
Cool. Questions, thoughts?
John, any feedback from your students on this one? How'd they take it from your experience?
So, I mean, just from informal, I haven't. So James, I actually haven't gotten the feedback from the survey yet, but at least informally, they seem to find it a worthwhile experience. When I asked them, I was like, this was their first time doing it. Did you find it useful? And they said they enjoyed it and thought it was pretty informative. So that's what I've gotten so far.
Yeah, John, I saw your email about that and thought that I sent it to you either yesterday or the day before. So,
Oh, okay. All right.
so let me know
I'll
if you
look
haven't
around. Yeah.
received it because I did see your you've got a bunch of feedback
Okay.
in the spreadsheet. So,
That's good.
so if you haven't,
Maybe I just missed it. I'm sorry. Yeah. So
no, no,
so
no,
I haven't seen that part yet.
I'm not. I just want to make sure I get it to you because I thought I had followed up, but I might not have. Um, uh, John, so what kind of like what do you have to like? Sometimes amphibians can be hard to identify, when you just kind of see them in the field without handling them. But is it mostly one or two types of things that you guys see at this site, or did you have?
Yeah. So there were I introduced them to the eight frog species I thought we were likely to see.
Uh-huh.
But I think they saw about 4 different species total. Most of them, I realized I needed to add an unidentified frog because a lot of them are just hopping away too quick for them to ID, but at least we get some abundance information. But I mean, some of them are obvious, like the bullfrogs are pretty obvious, leopard frogs. So they were able to get some IDs, but I would say the majority were unidentified. But the ones that are there are pretty distinguishable if you get more than a split second to look at them. Like there's also cricket frogs, which are much smaller and fairly distinct looking. We don't have a ton of species, so that's kind of an advantage, I think, in terms of being able to identify them versus like if you tried to do this in a place that had, dozens of species, then it's gonna be more challenging, but
Sure, sure. It also strikes me kind of hearing you talk about it, how, um, if there's at least in some field settings, if there might be an opportunity to sort of compare like along the riverside versus these backwater habitats, because I wouldn't be surprised if you know, you're going to see more of these and these particular kinds of amphibians in those kind of wetland habitats that are created off channel, and in contrast to the actual river channel itself, and kind of just highlighting the importance of that function of the river in terms of creating those, adjacent, ecosystems that are actually supporting a different, fauna than the river itself. Interesting.
yeah, absolutely. Yeah, I think that kind of contrast and thinking about the interconnectedness, and I did show them, we went through a map of like here's the river and here's where the marsh is relative to the river, and like
Yeah.
we're upstream of that. And what do you think, our activities in Peoria are having to this downstream marsh community? That sort of thing.
Yeah. Very cool. Anything else for John? Any other questions? All right, let's see who I got next here. Stream fish communities in suburban wetlands. Everybody guess. I'm going to go with Ryan. No?
That should be the second slide, though. Is there
Oh.
one more below it?
Oh, yeah. Sorry.
Yeah.
Is this,
Okay.
Yeah.
Yeah, there you go. So I ran this lesson in 2 different classes, different versions of it, of course, but with the idea that both classes could build a data set that would grow over time. So yeah, both classes went out twice in various ways. One class just went out twice formally, and the other one going out for a second stream sampling event was like their homework. So that helped me get more field grunt crew output out of them, which was very useful. We ended up sampling 5 sites total between the 2 classes during pretty much, I guess it would be October. And yeah, I also directed both classes to stream stats, where they had to delineate the watersheds where they sampled, and the assignments had to do with what the watershed attributes that might be structuring the fish communities could be. The students, as I knew would happen, had a really great time doing that. We had a lot of it just seemed like it's just a very engaging activity, which is why I wanted to expand this one. And we got a lot of fish diversity and a lot of sites that I hadn't surveyed before, because I decided to go through all the formal things and do this the right way. So in the previous slide, you can see all the organisms that we got. We got fully colored up rainbow darters in one site. The 2 on the bottom were in a Pittsburgh Municipal Park that took a little bit of extra emailing to get permission to go sample, but I'm glad we did, because we got some interesting things. Those 2 sites are in between. Well, one of them's in between 2 big ponds, like impoundments, and the other one is just upstream of one of those impoundments. So I think the fish communities are impacted by that. I don't have the assignments back from both classes, just one of them, but at least two students in the one where I got them back really connected well, in the sense that one of them pointed to watershed size being a major factor, which it clearly is in these data. So the Irwin Run isn't impacted. Like those data might look like it's impacted, but it's just small. And I didn't tell them that, but because I wanted them to find that on their own. And not everyone did, but at least one or two did. So that was good to see. So yeah.
And they're getting that metric from their StreamStat exploration,
They can
right?
get that metric from the stream stats,
Yeah.
but then they like kind of have to dive into the literature and because I asked them in the assignment what natural and human caused factors might be shaping these fish communities.
Mhm.
And that was the one I was looking for most, was watershed size. And so fortunately some people got it. And that the guy who got that one wasn't actually at Irwin Run, the tiny stream. So that was he somehow connected it in the literature, so that was good.
Mhm. Cool. Questions for Ryan?
Did you see
Ryan,
that over the, oh, sorry,
go ahead.
over the multiple days of sampling, like your students felt more confident the second time around?
Yeah, maybe subtly so. I think in both cases, there's enough of them there that like they're not too worried about doing it wrong or anything like that. But in the undergrad class, in the second one, yeah, they pretty much just kind of knew what to do and just dove right in. Even just wearing waders is like apparently a novel thing for some people. So that was
Most
something.
people probably.
Yeah, I guess so. Yeah, probably.
John, I see you've got your hand up.
Yeah, so these results are great. They look like classic biodiversity surveys where you have many taxa and you have a few that are really abundant and then a very long tail. Have you discussed biodiversity distributions with the class?
Yeah, that's so again, this was in two classes. The grad class, I don't have them, I'm not having them do any data work, and they come from different backgrounds, so I can't have them do figures like this. But in the undergrad class, these figures they're going to have to make on their own and turn them in, and they're going to have to comment on those patterns in the same way that you're pointing out there. And yeah, they've been introduced to that concept before, so hopefully they pick up on that. But I expect that they will, because they've already kind of been assessed on that in other assignments.
And so remind me, sorry, go ahead. Somebody else said something.
Oh yeah, I was just curious, Ryan, with this, because I know you had I think you put an emphasis on students using R for some of this. So did that part go well?
For one of the classes? For one of the classes, yeah. For the other class, for the grad class, they're not touching R. For the undergrad, yeah, they're using it. This assignment particularly isn't due until early December. So I kind of just accidentally gave them way more time than I should have, but I'm not going to undo that. So I haven't seen many of those turned in yet. In that class, they should be able to, I don't think it'll look this good, but some of them might get some figures that look close to this good. I'm going to have them calculate Shannon Weiner metrics and compare them among the 5 sites. And then next year, that'll be like, that'll go from five sites to 9 sites. So eventually I want them to do like explore some regression analyses with watershed size maybe and percent urban, because this is all a big suburban watershed. Each one of these drains into the same suburban watershed. So eventually we'll have enough data to show like hopefully some relationships in a regression framework. But I'm not making them do that now because I don't want them to get in their head that like 5 points is enough, which it isn't. So.
And this strikes me as one of those opportunities, and this is probably true for a variety of these lessons, but since you kind of brought it up with respect to that regression scenario that you're aiming for, is if other people do your lesson, then that can be another way that you could increase your sample size. And so I don't know if there are ways that you can. This is something we've talked a lot about in the network, and we haven't really solved it in a generalized way, but it might be something you could think about including in your lesson write-up. where, you know, you're kind of saying,
Oh,
hey,
yeah.
we're, you know, here's some example data from, you know, my systems, but, you
Yeah.
know, we're really looking to build this out. And, you know, I mean, you could, you know, do that regression. I don't know if you've got that regression embedded in the current lesson format or not, but you know, you could sort of say, well, we did this with 5 points, but we're really looking to build that out. So it's one of those things
Yeah.
that, like, the more people who do your lesson, the more valuable that shared data could become for the lesson itself.
Yeah, for sure. I think if I did that, I would maybe more incorporate biogeography into it, because if somebody did this in Idaho, you wouldn't see as many fish species, not in these small streams, right?
Yeah, you
So
might have
like
to
there
have
would be an
that.
element of like where are they sampling and what does that mean for the
Yeah.
numbers you would expect to see too?
Yeah.
Because it wouldn't be like 1-to-1, right? But it would be, but like sharing the data in the lesson, yeah, I didn't think of that, but happily will. And an invitation to like bring data together was also something that I would immediately put in for sure.
Yeah, you could definitely flag that. And I mean, I think what you've highlighted there is that, like again, one of those things that Cubes was originally designed for, but it hasn't, in my knowledge of the things that are on Cubes, I haven't really seen it used to the extent that I think it was originally envisioned. Is that there's supposed to be this kind of like forking of lessons and building of one lesson upon another, like taking some piece and then saying like, well, you know, I'm building on this portion of this lesson by making a new lesson. But you know, if you did find that there were other folks that picked this lesson up in Idaho, for example, then it actually begs that, you know, a second lesson that is more biogeographical in nature. So it could lead to, you know, maybe it doesn't make a lot of sense to include it yet with your data, but it might be one of those things that could become cool down the road.
Oh yeah. Yeah. If that were to happen, I'd be delighted.
Yeah. Cool. Other thoughts or questions for Ryan? Oh, next. Standardized protocols for data quality. This is, I'm going to guess Brad.
Yeah. Yeah. All right. So the lesson plan, again, the basic gist of it was that students would sample macroinvertebrates as a way to do a bioassessment, figure out essentially how healthy a stream is, whether it's impacted based on the level of tolerance of the different organisms that are found. So kind of had them do it on their own at first, just giving them some general guidance on sampling techniques. And then the lesson has them calculate a metric of their own design based on their counts of different organisms and the provided tolerances of each of those organisms. So they come up with a singular index that they think might represent the overall tolerance of that community on average. And then they calculated coefficients of variation as well as the average of each group's measures of that. They also collected some chemical parameters. It was just really quick to take a multimeter and have them record, you know, pH and dissolved oxygen and conductivity. So I had them get those really quick as well, so they could compare that to their invertebrate sampling. And as you would expect, they found much more consistent. Well, I'm jumping ahead of myself. I got them on the next slide. So hang on here for a second longer, maybe. Just what I actually did then. I had my 2 sections of an ecology class, 13 to 14 students in each of those. They were all biology majors or minors, sophomore through senior. They had the previous week was when Shaylee did her lesson with one of those 2 groups, and then I ran her lesson for the other group, which ended up working out pretty well because they kind of had this general introduction to getting comfortable in the stream, getting comfortable with the different kinds of organisms they might see, and then having a more targeted lab session where it's like, now we know the stream, and now we're going to try to carry out a highly productive activity, we'll say, as opposed to kind of exploratory activity. So yes, and after they got those initial numbers, I'm losing my trains of thought here. After they got those initial numbers, then they were instructed on a formal sampling protocol used by the EPA and compared their results. So maybe now go to the next slide. So the findings that I was starting to jump to initially, on the left there, that first picture is what they found after their first sampling period. And I wrote it on what I'm dubbing a Jabari board. So I brought a whiteboard out to the field.
I love it.
So as you might anticipate, things coming from a meter were fairly consistent. The confidence interval, confidence intervals, coefficients of variation on those was, from less than 1% deviation to 17% on dissolved oxygen. Probably had a lot to do with exactly where they sampled. But their bioassessment index using the macroinvertebrates was highly variable. The coefficient of variation was 142%, which is largely because of the one group who had a very high index that they calculated. But when I combined the two class sessions data together and I kind of trimmed off the most extreme values, it was still more variable than what they got when they used the formal procedure, which is shown on the right there. So when everyone used the same protocol, get the EPA protocol, the coefficient of variation was 8%, which actually meant it was more accurate than a dissolved oxygen meter, or more repeatable than a dissolved oxygen meter. So it gave the impression I wanted them to have, I suppose, that a well-designed protocol can lead to reliable results. And again, when I combined the two class sessions and trimmed off the extreme values for both indexes, I mean, you still had the coefficient of variation dropped to a quarter of what it was when they used self-designed protocols and everyone was doing things differently. It felt like a positive experience. There was cheering when they saw confident coefficients of variation, which is not a normal experience in a classroom. So they felt like they accomplished something and that they they were challenging themselves to do better than the first round when they saw their coefficients of variation looking not so good. The results, like I said, kind of showed what we wanted to see, which was that when you do things using systematic approaches, you get better results and more reliable results. Like I said, it paired well with Shaylee's activity as a lead-in. That was kind of a big thing that I'll have to think about with it. I don't think this would be good to jump into with a 3-hour lab without previous stream experience. I saw that the students had improved in their identification skills. I used ice cube trays for them to sort the invertebrates into little sections, and it made it very easy for me to just go scan them before they commit to their final counts and just find things that were clearly out of place to me. And I think it was notably less of a problem after they did the second round of sampling. So I think they started to cue in on what are the critical parts to look for. As everyone said, it was difficult to fit in 3 hours. What I ended up mostly having to rush through was the discussion as we went along the way. Some of that was they didn't need a ton of discussion. A lot of them are a little bit more advanced students, and so they kind of jumped pretty quickly to the kinds of thoughts that I would hope they would arrive at eventually. But it might be something that's better in a longer lab period or split between 2 lab experiences, perhaps. Maybe one that is kind of intro to stream and an initial sampling, and then a follow-up session later with a more formalized technique. And I think I just had the 2 slides.
Cool.
So I guess I'm done.
Thanks, Brad. Any questions for Brad? Oops, sorry.
Brad, was that cheering because they were excited to have sort of gotten the answer, gotten the result, or were they just like really thrilled about uncertainty and variation and stuff like that?
Oh yeah, no, we got a lot of central tendency and variation around the mean nerds in our group. No, no, they it was just the thrill of finally getting the number unveiled for, okay, here was our previous consistency level among groups, and the you know, our findings. And now finally here's the final result. And then there was just, you know, happiness that, hey, we did it. We met the objective of becoming more consistent and reliable in our data collection. So my classes here, they really respond well to gamifying and competition and stuff. So I think that played into their mentality pretty well of, you know, we're getting a PR personal record or something like that, you know, we're improving. And so I think for a lot of students that could be a fun element to kind of set that up as a challenge. Like, let's see if we can do better.
Cool. Excellent. Thanks for sharing. Next up, river temperature and habitat
There's
diversity.
Rachel's.
Right?
Yeah,
Oh,
said
this
she
is Rachel's, right? That's right. And so Rachel she wasn't able to join us. But she sent an update in an email to all of us that how it went for her. So, but just to be mindful of time, I'm gonna keep moving along then. Destiny, I suspect.
Yes, this is me. So you can see here my SLOs. I have them listed out. One that I added in and kind of reworded from last time was this SLO 4, which is helping them in real time develop an evacuation route from the place that they're in. And I came up with this because where I piloted my lesson was in a suburb. And as you can see, I piloted it with full ass adults, is what I call them. So homeowners, not college students. So people that have been out of college for over a decade. And a lot of them play in a band with my husband, so I had a captive audience, which is pretty cool. So we met in the suburb. The suburb has a second order stream that flows through it. And so we piloted the lesson in their backyard. So I think it went really well. One of the things that I kind of did anticipate, but didn't plan enough time for was everybody's excitement to see a floodplain map. I anticipated just showing it, helping them identify where they were at on it, and just moving on. And it ended up being like 30 minutes. So that was something that, again, like I thought, you know, people would like to see them, but not spend so much time on it. So that's a thing that I'm keeping in the back of my mind, especially for like supplemental material, is you know, helping people become literate of maps, but also being able to find that information. So like, you know, the homeowners are like, wait, where can I find this map? Because there were people from that neighborhood, but also some people that lived in like 2 neighborhoods over that came to the lesson. So that was a big thing for me. Another thing that I kind of knew already going into it is that 120 minutes was what I want to do this lesson in, and I tried to do it in a compressed version of that. I need a lot of time, especially if you're going to spend 30 minutes talking about a map, which is just, you know, my first SLO here is like, where are you located in this floodplain? It needs more time, and maybe that means maybe removing one of these or breaking it up into 2 lessons. What does that look like? I'm still ruminating on. So more time, always more time. Also with my lesson, I've created a booklet. I love booklets. I've done a lot of K-12 education, and kids love booklets. I love booklets, and apparently homeowners love booklets too. So I designed a little booklet to go along with this activity where they could just keep up with all the different things that we're doing. And for SLO 1, 2, and 3, I have them doing sketches of the floodplain that they're standing in, but I was having them redraw the floodplain each time. And that was like one of the biggest like feedbacks that I got in real time piloting the lesson was like, have one sheet where we can sketch and add things in, and then bring different color pens so that when we move from SLO 1, I could do it like blue pen. SLO 2, I can do it in pink. SLO 3, I can do it in red. And at the end, I have one composite picture of all of the things you want me to identify as these flood risks. So streamlining that little booklet is something that I've been working on, and especially again with that sketching part. But overall, I felt like it was great. It was funny. I wish I would have taken pictures, but October 25th was Saturday, or was it a Friday? It was a Saturday. And the band was playing a Halloween show. Everybody was in costumes. just hanging out, playing in the stream. It was absolutely excellent. But I was like, you know, in teacher mode, not thinking about taking pics. But it went well overall. I am excited to see what their reviews were, because they did give me a lot of stuff that I just wrote down for myself while I was there. And they took notes on their little booklets that I'm going through. But I'm excited to see what their results were for the feedback.
Destiny, when did you pilot?
October 25th. So not very long.
Yeah. Yeah, I'll check in. They might be some new results in there since I last looked.
Yeah.
I think I did see at least one there last time I looked. It occurred to me that unfortunately he was not able to join us today, but I bet Nico would have some interesting thoughts about that, sharing a flood map with people, given his project. I wonder. Like, he's created some really fun graphics for, you know, because for the cheat watershed, since he's in there, which some of you, you know, we all saw when we were out at the cheat. But I wonder how he approaches that, and what kinds of things that he you know, how what kind of responses he gets, if that's something that he also finds that people really like nerd out on and really like intrigued by.
Yeah, totally. Yeah, I reached out to him before, and he did look over my general lesson, but asking him specifically about that, I think would be a very good idea, especially if he has all those resources already compiled. Just including them as a supplemental material section for this lesson could be super helpful.
Yeah. Very cool. Other questions for Destiny?
Yeah, do you think this will work with not full ass adults?
I do. I think that all of the components are super important, especially for people that are teaching on a campus that has a stream running through it of any sort. Especially thinking about, you know, all of those the features that are there. So getting them used to the morphology of the channel, thinking about the dangers, how high floodwaters have gotten in the past, and this idea of the evacuation route, that's super important. The neighborhood that I piloted this in, since Hurricane Helene, only has one road in and out, and every time it rains, that road floods. And so they were thinking like, holy crap, like actually my evacuation route needs to happen 30 minutes into the rainstorm, or I'm climbing up the mountain with my stuff on my back. So I think, you know, it's definitely applicable to any place that has a stream, and with any audience.
John, I see your hand.
Yeah, so you mentioned Hurricane Helene. That should probably be the dominant signal or dominant event that leaves flood debris or evidence of it. Did you find evidence preceding that or since then? Or is it just Helene?
So Helene is specifically in this neighborhood where I went, the biggest thing that's impacted that neighborhood. So when we were thinking about specifically SLO3 here, everybody pointed to the fact that the bridge is gone, the only bridge that was in the neighborhood is gone. So helping them identify that, that was really important. But, you know, we also noticed, and I made them look for smaller scale impacts, right? So how can we tell that a rainstorm might have happened 2 weeks ago, leading them to identify like where the leaves are on the bank itself. So yeah. Oh, I think my internet is going bad.
Awesome. Thanks so much.
Yep.
Lisa, I'm guessing.
Yep, that's mine. So I took 5 students from my environmental geology course out to the field. It was a big course. I didn't want to take the whole class out. And so I kind of took volunteers. And so we went out, we kind of did a description of the riverbank itself, and kind of see the picture of the students kind of looking with their hand lens, looking at the sediments and describing them. And then I had the students take images with iPads. I provided the iPads for them so that they could get these 3D images. And I was thinking I should put that up here, but I didn't get that shared. And then we kind of talked about some of the different processes. And so where this fit, it did tie into this information that the topic I was talking about leading up to this in the class. So we had talked about kind of different scenarios of erosion, and we had talked about case studies of restoration too. And so the students had a little bit of that information before we went out to the field. The challenges, like everybody's talked about, there were challenges with time, partly related to just kind of getting out and on the road and getting to the site, and like getting from the parking area to the site where we worked. We had a little bit of a walk into this particular stream site. And then there were different parts of it where I feel like in some ways, because this was a small group of students and it felt a little bit more informal, that some of the conversations, some of the reasons why time was a problem was because our conversations got sidetracked. But at the same time, I think if I would have had a big class of students, we probably still would have had challenges with the time, because then there would just be more people to kind of keep on task. One of the lessons I learned was kind of constraining students to where they went, because I kind of gave students the tools and let them go. And some of them went much further along the creek than I anticipated them going. And so they kind of wandered off a little bit. So kind of keeping them kind of reined into specific sites so that it's easier to bring everybody back together for our discussions, I think, is one of the things. Another challenge that I had was kind of going into it thinking I had a really small group and we have some equipment for students to get boots so that they would be able to walk in the stream, and getting out there and realizing that things didn't fit the way that I expected them to. That I guess kind of brought up a little bit of an accessibility. How do we make that equipment work for all the different shapes and sizes of people? But the other part, I think, a really good thing about it was that the students from like the beginning to the end were really in a good, were really positive, really engaged in the activity. They asked a lot of questions, and then that was also part of the challenge with the time, is that we really got into some good conversations as we were working, but that kind of, and so it was a good experience, but kind of a couple of different areas where I think we can tweak the activity here.
Very cool. Thanks. Questions for Lisa? It makes me think of that activity we did with Anita, Lisa, when you were here for the field safety workshop. And just kind of, because I actually, the waiters not fitting, I've heard that being actually a common kind of like issue in terms of making people feel welcome and making people feel like they belong, and kind of like not the person that doesn't fit into the waiters, and that can be an issue. Although, I mean, there may be a variety of ways to do a workaround with that, but I almost wonder, like, how she had us kind of do that sort of away team versus home team with the using our phones. I almost wonder if you could do that with people who want to wade and those that don't. I mean, if they're really close, then maybe it's kind of silly. I don't know. You can just talk to each other. But anyway, it's an interesting thing to think about.
Yeah, we did end up having to do a little bit where the ones who the waiters worked for them, they went across the stream and they did more of the distal kind of imaging, and then kind of shifted gears so that the folks that weren't going in the water did the other side of the river that we got to with the trail. So we did kind of break it up into different tasks, different duties with that. Yeah.
Yeah, I'm hoping Anita will come for the rendezvous next year in Oklahoma. Other comments, questions? Thanks, Lisa.
All right.
And Matt. I think Matt had the benefit of.
I get the bonus for putting my name on there.
You do. Extra 5 points for Matt.
All right. So yeah, thanks. I think I'm the last one here, so I'll try to keep it quick. I was able to do mine 2 different times, 2 sections of my ecology lab, 18 to 20 students. It's a 300 level class, but really it's an intro level ecology class. The objectives here were to really get the students to think carefully about microhabitats and the tiny organisms that live in aquatic systems and how, you might be able to walk a few feet and you're suddenly in a new part of the system that might have different organisms or at least a different composition. So we had them generate hypotheses in class. Then I took a page out of Anita Marshall's playbook and did split up the group into the field team and mission control. In this case, it was part field sampling. So we decided what we wanted to do. For my classes, they mostly stuck to flow differences. So like high versus low flow. Mission control had to set up microscopes and was responsible for cleaning everything at the end of the 1 hour and 50-minute lab. So it's a pretty short amount of time. We hopped in the van with the field team and went down to this beautiful field site on the Appomattox River and sampled in a couple different spots. Then students came back, had to make a wet mount. Every student had to do that, put something under the scope and then draw it. And then we collected data along the way in both sections. And then in the second week, we followed up with a brief on paper analysis where we built rank abundance curves and talked about our hypothesis. In my lesson plan right now, I have them writing a protocol for how to do this. And that was a hat tip to Susan for that one. And I like that. I used it again in a different lesson and it worked better. Some of the students just didn't really do the protocol, but everyone in person was reflecting well on the hypothesis. So I'm going to fold that in as a deliverable instead of the protocol for this one. Where that worked well was when my students had to write a protocol for another class and the class actually used the protocol. So I think because they felt a little ownership over that, they did a nicer job. In general, this went really well. Time is always of the essence with an hour and 50-minute class, but splitting the group into 2 teams really helped make that happen. And each person, whether they came in the field or not, felt valuable, I think. And we got some nice shots of things under the microscope, like this desmid. This is the first semester that we haven't found tardigrades, so big disappointment all around. But I snuck a few into the data set. I sort of had a little Wizard of Oz control there. And so they're in the rank abundance curves that you see. In general, I'm totally going to do this again. If you had a 3-hour lab, I think you could do everything. But this is now folded into my curriculum for my ecology lab. So it's there.
Awesome. Questions for Matt? Matt, quick question. So remind me, if your students, is it really important that they know the organisms, or is it that once you sample them, you're basically like making them into morphospecies as you identify them? And
yeah,
how does that work for the lab?
Yeah, so that was another challenge I forgot to mention, was that we don't have a lot of time to sort of discuss the tree of life and to, we're looking at very different things. Some things are algae and different clades, and some of these things are multicellular animals, and there's protists. I don't get too freaked out about that. What I do is I have them share with each other, take photos, do Google Lens searches and INAT, and see how close they can get. And then I give them some names to kind of push them along. But this lab does not put an emphasis on proper identification or phylogeny or anything.
Yeah.
That's an area where this could grow. And so for us, it was just like, see what you can find. How many, sort of needles in the haystack can you find? And when we find something, we'll explore and try to identify it. But that is a challenge to this lesson, is if you go in without much knowledge, you're sort of wondering what you're looking at, but that's part of the excitement too. You want to know what it is.
The gateway to entry is pretty minimal. I mean, you don't have to have already built up like a bunch of understanding of these aquatic microinvertebrates, which a lot of classes won't have, right? So you can,
That's right.
it's the discovery is what is kind of what you're selling in a sense, almost. Yeah.
Yep.
Cool.
Yep.
I can see it. Yeah. Very cool. I can see it be applying, lots of different kinds of aquatic ecosystems. Yeah.
Yeah, and I did do this on a stream bank, but I collected samples in a different lab from a little pond on campus. So it's very transferable, the lesson, to any place that has some water.
Yeah. Yeah.
We could even do it. Who was I talking to at the rice retreat there about? Could even do it with, like, if you're in a pinch, you could use the liquid in a worm bin and find cool stuff in there. So if you can't get in the field, there might be ways to do it.
Or rock pools.
Yeah, even better.
Yeah. Very cool. Thanks for sharing. Any other questions for Matt? Awesome. Well, we have all run a bit over, so I'm going to go ahead and bring us to a close. Thank you for sticking around and hearing from each other. I'll post the folder up for people to start putting revisions in, and make sure that you get a reviewer to take another look at your final, as you're heading into the final home stretch. I'll touch base with Susan to try to figure out a good day to do the This is Cubes onboarding. And thank you so much for sharing. You guys are doing some really cool stuff, super diverse, new stakeholders being engaged, dozens of students being engaged. It's really exciting to see. You guys have a good rest of your Friday. Enjoy the weekend.
Thanks, everyone.
Take care, y'all.